chair for the session today to introduce our speaker. Sarina, can you please?
Yeah, hi everyone. Welcome and thanks for joining. It's my pleasure to introduce Anna Heath, who is a scientist at the Hospital for Sick Children in Toronto and who leads the Innovative Methods in Pediatric Clinical Trials Impact Lab. Her research focuses on this developing statistical innovations for clinical trial prioritization and design, particularly focused on Bayesian methods and decision analysis. She is also the lead for the Statistical Analysis Committee for the practical trial, and I can tell you that none of what we've accomplished so far would be possible without her expertise. So, Anna, I'm actually really excited to hear your talk, and we'll let you take it away.
Great, thank you so much, and thanks for this invitation today. Let me just share my slides. Perfect. Okay, so I'm going to be talking today about designing an adaptive platform trial, a statistical perspective. So as Sarina said, my research focuses primarily on developing new statistical methods for clinical trial design. So that's really the perspective that I'm going to kind of be talking about today. Firstly, I have some disclosures. I don't have any industry disclosures. I hold grants from these various research councils. I have a strong bias towards Bayesian methods for clinical research. So I'm going to be talking about Bayesian methods for platform trials. There are non-Bayesian methods for platform trials, but I don't understand them, so I won't be talking about that. And as Sarina said, I lead the Statistical Analysis Committee for Practical. So while the methods that I'm going to be talking about are more general, the kind of examples that I'm going to be talking about will come from Practical. But that's just because they're the ones that I had at my fingertips. Okay. So the learning objectives for today are just to discuss some key considerations specifically for Bayesian adaptive platform trials. The first thing I'm just going to talk about is an introduction to Bayesian interpretation of clinical trials and some of the kind of key concepts that we have in a Bayesian analysis. I'm going to talk a little bit about different types of adaptive trials, and in particular this concept of dynamic borrowing, which we use in practical specifically, but it's also kind of a tool that's been used across lots of different platform trials to kind of gain efficiencies from the platform kind of structure. And then finally, I'm going to be talking about kind of simulation-based assessment of clinical trial designs and just talk through kind of, I guess, like a framework, or at least a framework that I like to think about when we do these simulation-based assessments. And hopefully this will give you a bit of a flavor of what the statistician does in the background, but also maybe give you some kind of tips and tricks for how to kind of, I guess, interact with that process of simulation-based clinical trial design. And obviously, for those who are statisticians, hopefully it rings true with what you've also seen. So just to start with a really kind of a nice introduction to Bayesian methods. I think one of the things I like to think about Bayesian methods is that actually there's a lot of like simplicity in kind of the ideas and what actually underpins the machinery. Of course, we can layer as much complexity as we want on top of that. But to my mind, the ideas that underpin Bayesian methods are actually simpler than a lot of what people are used to thinking about in the kind of more traditional frequentist methods. So when we start with a Bayesian analysis, the first thing we start with is what we call a prior distribution. So a prior distribution it's just a way to conceptualize what we believe before we look at the data in the trial. So in this particular case, I've got this distribution here. It's for a probability. And what we're saying with this distribution is that we think before we look at the data that the probability lies somewhere between 0.1 and 0.8. So I think we can probably all agree that that's a pretty like vague thing to believe, like I think in most kind of processes that you might be more familiar with in your clinical work, you would actually have a much better idea about where that probability lies. So it may be the probability of somebody being taken off ECMO. You'd actually have a much better idea of that than between 0.1 and 0.8. But nonetheless, we can start with this kind of prior distribution. And then what we do is we actually collect the data. So this is, I guess, there's nothing Bayesian about collecting data. We collect the data, and then we conceptualize it in this kind of distribution that says that if, based on the data that we've collected, we would think that this probability lies somewhere between about 0.4 and 0.8. So the data has kind of given us some extra information, and it kind of allows us to update our belief. And so what we have here is what we call the posterior distribution, and it's essentially a compromise between the prior and the data, and that allows us to kind of understand both what we did before and what we currently saw in the data, and give us this distribution that represents everything we know about this quantity of interest. In this case, say the probability that we're interested in. And as you can see, as you kind of the posterior distribution in this case kind of matches pretty closely to the data. So it's actually the data that's really driving what we believe about the parameter, which makes sense because before we looked at the data, we were really uncertain about what that true probability is. So I think, to my mind, these are the three concepts that you kind of need to understand for a Bayesian analysis. We have a prior distribution, which is what we believed before we looked at the data. We have the data, which is what the data says. And then we have this concept of a posterior distribution, which is a combination of those two pieces of information. And once we have that, we're actually kind of done, that's the analysis. That's everything that can be learned about the parameter from the data that we have. And what we say in a Bayesian analysis is that the posterior distribution, this kind of summary of the data in the prior, contains all the information that we might have about the treatment effect. So in a clinical trial, we calculate the posterior of the treatment effect, and that contains everything that we need to know about the treatment effect. What we can then do with that, and this is what's more commonly done, is we can summarize that posterior distribution in different ways in order to be able to kind of understand or interpret the information that actually came from the trial. So for example, we might represent the posterior by a mean, and that would be the kind of the average treatment effect or the best guess of the treatment effect based on the data in the prior distribution. We can estimate the median, which usually is fairly similar. In this case, it's slightly lower because we have a skewed distribution for our odds ratio on the right-hand side of the screen. We can calculate the variance or the standard deviation of this posterior distribution, and that would be equivalent to kind of calculating the standard error in a kind of frequentist analysis. So that would essentially gives us some idea of the variability or the uncertainty in our estimate of the odds ratio. We can also calculate what we call a credible interval, which essentially is just rather than having like a single best guess for the estimate, it gives us a 95% probability that that estimate lies between 0.94 and 2.56. And I think in a lot of ways, this is where the Bayesian analysis actually allows us to say what I think clinically often people really want to say, which is what is the probability that my treatment effect is bigger than 1? There's a 95% probability that my treatment effect is between these two values. And that's exactly what you can say in a Bayesian analysis. And I think that's why it's very powerful as a kind of more as a communication tool. And crucially, what we can say, as I just said, is the probability of a positive treatment effect. So in this case, we have an odds ratio. If the odds ratio is greater than 1, then we have a positive treatment effect for this intervention, and we can calculate the probability of that. So in this particular case, it's 0.98. As you can see on the right-hand side of the screen, the majority of the posterior distribution is greater than 1. And this is something that I really think is one of the most interpretable ways of kind of understanding whether you actually have an effective treatment or not. And so that's really why I kind of prefer this type of analysis, because that just makes it kind of easier to make those interpretations and to really understand the probability of a positive treatment effect. And what you actually do with that is that something that allows you to use that intervention in practice. In a lot of ways, it depends on the other profiles of the intervention. For example, is it cheaper? If it's cheaper, then maybe the probability of a positive treatment effect being 0.98 would mean, okay, we're happy to use this. Does it have lower side effects? Then maybe we'd be happy to use it. On the other hand, if it has much more side effects and it's much more expensive, we would be maybe a bit more hesitant that there's a probability of a positive treatment effect. Or maybe we'd want to see not just a positive treatment effect, but it being significantly greater than 1, say 1.2 or 1.5. And again, the posterior distribution can just be summarized in that way to give that information. So it's a really powerful tool to kind of give you the output that you're interested in. So that's all I have to say on Bayesian analysis. I sometimes like to just stop there, mainly to let people digest, but also if there are any questions about Bayesian analysis at this moment, I can also take them now. Okay, fantastic. So I'll assume everybody's understood everything they need to know about Bayesian analysis, and we're all on board with this being the best way to analyze trials. Perfect. So what we were actually, I guess, talking about today and is, I guess, taking this Bayesian concept, which can be used in kind of any trial design that you want, but actually we're starting to think now about, I guess, adaptive platform trials. So now we're not just thinking about how do we do a Bayesian analysis, but how do we adapt a Bayesian trial? So the FDA defines an adaptive design as a design that allows for prospectively planned modifications to the design based on accumulating data from the trial. I always like to put this definition up whenever I'm talking about adaptive trials because I think it's really critical to reflect on this. The key thing with an adaptive design is that before you do the trial, you've already planned how you're going to modify the data. Modify the trial, sorry. So it's not that like you just decide what to do halfway through. You already know exactly how you're going to adapt your trial based on what data you observe. So it's really almost like a recipe where you collect the data and then depending on what it tells you, you go down these different pathways. So the key goal of statistical trial design is to determine which adaptations should actually be made. So there's a huge range of different adaptations you could use, and I'm going to talk about a few of them in the next few slides. And not only which adaptations should be made, but the impact of these adaptations. So we're not just going blind and being like, yeah, let's hope that this will be fun, but we actually understand how these adaptations are going to affect our trial conduct. And some of that might be, I guess, like traditional measures of trial success, like power or type 1 error of the trial. But other kind of goals of these statistical trial design might be to understand what happens if the recruitment rate is slower than we're expecting, or what happens if the effect that the intervention affects people in a different way than we were anticipating. Can we detect that? Do we understand what we're doing? So it might be, again, as we go through the presentation, I'll probably end up focusing more and more on power and type 1 error, but actually statistical trial design can be much broader than that and really focus on whatever's relevant to the clinical team. And the crucial kind of thing here is that because these adaptations have to be prospectively planned, essentially what we're determining is what we call decision rules for the trial adaptation. These are kind of the rules of how we're going to adapt the trial as the data appears in the trial. So it's something that's actually fixed in advance of the trial and allows us to adapt as we go. So there are different types of adaptive trial. I think I always like to highlight the first one. Dose-finding studies are actually probably the most common type of adaptive trial. A dose-finding study is where it's even the most simplest dose-finding study where you do a 3+3 design, you give the drug to 3 people, you see how many toxicities they have, you either escalate or stay the same, is exactly an adaptive trial. You're giving the drug to 3 people and then you're seeing what happens. And if that goes well, then you escalate the dose. If not, you stay at the same thing. So actually, people are very comfortable with adaptive trials in that dose finding, dose response context. But as we go more into the kind of later phase trials, we've become less and less confident. There are other types, and the most common, I guess, type that we use in these kind of more bigger platform trials are what we call kind of sequential monitoring. So where you're looking at the data in the trial as you go through, and you're trying to see whether you can conclude efficacy of the intervention. So which intervention is best, futility. So is it going to be not possible to determine whether an intervention is better? equivalence of the interventions and things like that. So really, we're just looking at the data and we're trying to see, can we make a decision about which intervention is better or worse or the same as we go through the trial. Another is this idea of response adaptive randomization or covariate adaptive randomization, where you're trying to kind of adapt who receives which intervention based on either their response or the covariates in the trial. So maybe you initially recruit too many men, and then you look at the data and you say, okay, well, we need to kind of increase the proportion of women in our trial going forward. So we're going to try and boost that recruitment and that randomization. Another key adaptation is what we can call an enrichment design, which is where we kind of try to focus the trial population on people who are more likely to benefit from the intervention. And then another kind of key type is this idea of a seamless design, where you can potentially move from different kind of research questions with the same intervention. So maybe you start with a kind of short-term feasibility outcome or even a short-term efficacy outcome. And then as you determine that there's maybe that intervention does work on that short-term efficacy outcome, we kind of seamlessly proceed into a bigger, more expansive trial with maybe a harder or a more long-term outcome. And one of the advantages, mostly the advantage of that, is that you can potentially kind of include the patients who have already been enrolled in that feasibility portion of the trial and kind of roll them forward into the main bigger trial and actually reduce your kind of sample size requirement going forward. So there's lots of nice things. And I think in terms of platform trials, we can use kind of all of these four kind of adaptations that I've said, those last four. The sequential monitoring is very common. Response adaptive randomization is sometimes used. I think enrichment designs are becoming more common, and certainly as you have these broad patient populations that come into your platform trial, you might look to enrich and only focus specific interventions on specific kind of subpopulations within that trial. And then again, the seamless design is really key for the platform that allows you to sort of use the operational and the administrative kind of effort that you've already done in setting up the trial on the platform and roll that forward into maybe a bigger comparison as you go forward. So I wanted to just focus a little bit more on some of the concepts of this kind of idea of sequential monitoring, because I think that's actually the most important kind of setting. So essentially what we have here is we essentially have a representation of the relative effectiveness of an intervention on the x-axis. And essentially we have this blue line at the null effect. So that effect might be 0 if you're looking at like a risk difference or a mean difference, or it might be 1 if you're looking at an odds ratio or hazard ratio. And what you're looking for are these different concepts. So I think probably for me, the simplest or the one that's most standard would be this idea of superiority. So you're looking for something bigger than the null effect, better than the null effect. And the converse of that might be inferiority. So where you have something that is worse than the null effect. So you might be looking for inferiority of an intervention if you have multiple interventions in your platform. So maybe you're comparing 5 different interventions. You might look for inferiority of that intervention compared to all the others. And then that would allow you to reduce the number of arms in your trial to say 4 or 3 if there are more inferior interventions. Equivalence, I think, is I guess kind of, it's essentially this idea that they have the same effect. But statistically, we can't say, do they have an effect of 0 or 1? You know, do they have exactly the same effect? So we have to create these kind of bands of equivalence. So where we would say, okay, well, within this band, we don't mind whether the interventions differ very slightly. And the bigger you make those bands, the easier it will be to declare equivalence. But obviously, the less you could say that they're actually equivalent because they end up being fairly different from each other. And then futility, I think for me is probably one of the hardest to kind of conceptualize. It's not necessarily that the intervention is worse, i.e. inferiority, but it's that it's not much better than. It's kind of equivalent to, I guess, like a non-inferiority trial. It's this idea that like we think that it's either worse or it's not sufficiently better to continue with the trial. So that's really where I guess you have this band for equivalence. Futility is kind of like inferiority and equivalence at the same time. So it's roughly the same as the intervention. And we might use that if we had I guess the standard of care was part of the trial, and we're really looking for something to be much better than the standard of care in order for us to kind of go through with the effort of changing clinical practice. And so futility would be there at that point where you'd be like, okay, well, this is a novel intervention. If it's futile, it just means that it's not sufficiently better than the standard of care. Oh, hello. Okay, perfect. And just to kind of give you, again, to bring it back to this Bayesian concept, essentially what you're going to have, and maybe I can, okay. Essentially what you're going to have is you're going to have a posterior distribution that kind of is on this axis of relative effectiveness. And what we can calculate is we can calculate the probability that they fall in each of these categories. So the probability of superiority, the probability of utility. And if your posterior distribution lies very much in this superior part of the distribution, you're going to get obviously a high probability of superiority and a low probability of these other kind of categories. And so we will be able to use that probability of superiority, futility, etc., to make decisions in our trials. So, for example, if we have a high probability of superiority, we might say that we can stop the trial and move on and conclude that the intervention is superior. So that's essentially how that Bayesian analysis intersects with this idea of monitoring. The other kind of key concept that I just wanted to talk about, because I think it's becoming more common, is this idea of an enrichment design. So what we have here is I have a green line that represents the response probability that's stationary across the covariate values. So we might have a covariate here that's to do with like some kind of probability. It's on a 0 to 1 scale, and the response probabilities of the probability of death, for example, might be the same across all that covariate value. Or alternatively, you might have an intervention where it varies. So for low values of the covariate, you might have a lower response probability, so a lower probability of death. But as that covariate increases, you see that the probability of death increases. And so if we were to analyze all these patients all together, we would actually end up potentially concluding that this treatment had a null effect. So the people who have high covariate values balance out the people that have low. But if we can identify instead to focus only on the kind of green section of the covariate values, then that would allow us to kind of A, detect that there is a positive effect, and B, essentially avoid harming the patients who were in that higher category. So an enrichment trial or an enrichment design would basically adaptively try to find this green section of the graphic. So you're essentially going to start by enrolling everybody in your trial, and then as you go through the trial, you're going to sort of analyze the data and try to estimate this kind of line here. And as you estimate it, you can identify this kind of cut point and focus on this green side of the graph so that you can really focus on the patients who benefit from the intervention. And I think that as we go through, this is something that, as I said, is becoming more common in platform trials just because we have such a broad inclusion criteria. Okay. So the other kind of key element that comes in a lot in these platform trials is this idea of dynamic borrowing. And essentially, Bayesian methods kind of allow you to do this in maybe an interpretable way. And essentially, it's to do with this idea of heterogeneous treatment effect. So again, thinking back to this idea in these enrichment designs, where we might have a lower response probability for some covariates and a higher response probability in other covariates, that's what we're trying to detect with this kind of, or trying to investigate with this dynamic borrowing. So we have this kind of covariate that we're using in some way to separate these different states. So these different patient states or patient kind of groups or subgroups. And we can, in a kind of standard framework, we could either analyze these subgroups kind of independently from each other. So we're going to get a different treatment effect in each subgroup. And again, that treatment effect might be heterogeneous. It might be different in each subgroup. Or alternatively, the standard approach might be that you would just analyze all the subgroups together. So instead of accounting for different potential differences, we just analyze them all in one kind of setting. And the dynamic borrowing approach basically allows you to kind of rest somewhere in the middle. So it's not quite a pooled analysis and it's not quite an independent analysis. And essentially the goal is that we can learn from the data how similar these patients are to each other, and so how close you should be to a pooled analysis. So you can think about it as like kind of a compromise between this independent analysis and this pooled analysis, where the data tells you where in that distribution you should actually lie, or where on this graphic you lie. And again, this is a Bayesian analysis. So what we actually have is we have essentially a prior distribution that tells us what we currently believe is the kind of where we should sit on this spectrum between independent and pooled analyses. And then we update this with the data, and that may move us kind of further towards an independent analysis if we're seeing big differences between the different treatment groups, or closer to a pooled analysis if the differences between the treatment groups are quite small. And the advantage of this is that when you kind of borrow, you actually get greater precision on your treatment effect estimates. So your posterior distributions have lower variance, which again intersects with this idea of sequential monitoring. It allows you to potentially conclude superiority or futility faster, because you'll have more probability on either side, either the superiority side or the futility side. So that's really where we get this kind of benefit from in a Bayesian adaptive trial, is that because we're putting all of these patients together in this platform, we can actually analyze all the data together and understand whether it's appropriate to keep those states or those subgroups separate or pooled. And it allows us to kind of statistically combine the information. So those are, I guess, some of the key concepts in kind of Bayesian adaptive platform trials. There's different types of adaptation, and then this idea of borrowing, which allows us to kind of provide statistical efficiency from the actual design that we have. But the next kind of piece of the puzzle is, how do we actually do this? So we have kind of all these great ideas about how we can design our trial and what that can mean and what that can help us identify statistically. And we need to understand how that affects the design. As we said, kind of statistical trial design in this context is to understand, firstly, what adaptations we can do, but secondly, how does that affect the trial design? So we need to design these decision rules for the trial adaptations. So we know what type of adaptation we're going to do, but we need to understand how we're going to make those decisions as the trial progresses. And we call this, or often call this, like a trigger for the adaptation. So we know that we're going to have this adaptation, and we want to know what is the trigger, what is the kind of threshold or the analysis that we do that says, oh, okay, right, we need to do this adaptation. And the effect of different adaptations need to be compared, so we can choose kind of the optimal design for our trial. And I think that's one of the things that makes, I guess, these adaptive trials much more complex than a standard trial, is that usually the main thing that we're considering in a fixed design would just be the sample size, and that will be affected by the budget and things like that. But here we could do a huge number of different potential adaptations, and we need to know which one is the most appropriate. Some of that will be the most appropriate based on the clinical needs of the study, and some of that will be to do with the statistical considerations that you need. So there's a lot of that. And because these adaptive trials are very complex, we often require simulations. So in a fixed design, you can often use kind of formulas that allow you to calculate the sample size analytically, which means that you can have like an online calculator that does it all for you. But in these adaptive trials, there's really no way to do that analytically, or it's very rare to be able to do that analytically. So we have to do these simulations. And what I have on the right-hand side of the screen here is just kind of a picture of some of the simulations that we might do. So what you have is you have the treatment effect on the y-axis and the number of patients on the x-axis. So what you can see is that we're running the trial from 750 patients to 2,000 patients. And when the treatment effect reaches a certain level, a certain trigger, that's when we stop the trial. So that's why you have some of these shorter lines, because that's where we only simulated, say, 800 patients or 850 patients. And what you can see is that like some of the lines are colored. So in this particular case, what we have for the pink lines, these are studies that stopped for efficacy, so superiority of the novel intervention. And the green lines are those that stopped for futility. So you can see that as the treatment effect is estimated lower, they're more likely to stop for futility. And if the treatment effect is estimated higher, they're more likely to stop for superiority. So that's essentially what we're going to do. We're going to run in the computer lots and lots of patients, kind of pseudo patients, and we're going to track what happens to the trial as we go through with our adaptations. So how do we actually do that? How do we set that up? And one of the things that I think is maybe challenging about setting this up is that, of course, this isn't just a statistical exercise where I get to sit in front of my computer and code and live my best life and then run the simulations. This is something that has practical it needs to be run in reality. So really we need to have a conversation and communication between the statistical team and the clinical team to make sure that the adaptive design and the platform trial respects the needs of the clinical investigators, but also respects the statistical kind of constraints that we have. So this is how I think about designing a simulation. Over the years, I've looked for a really good paper on how to design clinical trial simulations, and they're coming, but I haven't seen the one that I want to cite yet. So this is the way that I think about it. So the first thing is to specify the research question. So that is the PICO. Obviously, we need to know what is the clinical trial that we're interested in. But the other key thing is to really understand the goal of the adaptations, the goal of the adaptive design, because as we said, there's lots of different types of adaptive design. And so really what we need to think about is what do you want to achieve with your adaptations? Do you just want the opportunity to stop early if you've got a sign of efficacy? Do you want to look for kind of covariate adjustment and seeing whether there's a group of patients who would benefit more? Do you just want to kind of give yourself a little check in case the baseline risk in the control arm is much lower than you were expecting, and so actually you're going to need a larger sample size than you thought? And these are all like different goals that require different designs. Then you need to develop a model for your data analysis. So how are you going to analyze the data in the trial? But also that kind of gives you a model for data simulation. So in this case, we need to work out how we're going to simulate the patients and what they look like. We have to design the trial decision rules. So what are those rules actually going to look like? How are we going to make changes to the trial conduct as the trial runs? Then we need to determine the key scenarios for simulation. This is really where I guess the key kind of interaction happens between really now we've got this statistical design, but we need to understand what are the relevant scenarios from a clinical perspective in order to design the trial appropriately. Then we run the simulation, and that can be quite an in-depth process, sometimes weeks, sometimes months. So it's not, a nice 5, okay, we've done all of that and now we can do everything in a few hours. It can really take a little bit of time. And then, of course, the main goal to evaluate the results and present to key stakeholders. And often the kind of 4, 5, and 6, or even 3, 4, 5, and 6 will kind of iterate. So you'll present the results to the key stakeholders, and then that might suggest that you change the trial decision rules and you go back through 3, 4, and 5. So to give you an example of how this actually works in practice, I'm going to give an example from the practical platform trial. Usually when I present this, I present to people who aren't quite so expert in acute hypoxemic respiratory failure. So apologies to all my clinical collaborators if I make mistakes in the context. But this is our PICO question. So we have patients with acute hypoxemic respiratory failure with other kind of restrictions on their elastin state and what respiratory support they are receiving. The intervention is a novel ventilation strategy, and it's being compared to the standard of care. And the outcome is this ventilator-free days, which is a common outcome. We wanted to have these kind of 3 adaptations, mostly for futility and efficacy, or futility and superiority, but we also allow ourselves to adapt for treatment effect heterogeneity. In particular, we're looking at pre-specified heterogeneity. I'm going to focus the design here on this trial 3. So we have patients in 2 different severity states, and we're going to make decisions separately for each severity state. So that will allow us to say that the treatment is effective, say in severity state 2, but not in severity state 3. So again, we're kind of doing a kind of a, I don't know what the right word is, like a crude evaluation of treatment effect heterogeneity. We're not going the full way and evaluating exactly where maybe the cut point is for that heterogeneity, but we will allow ourselves to say whether it works in one state and not the other. So that's our PICO and the kind of adaptations that we were hoping to make. And then we needed to find a model for the data. So here on the bottom, I have this ventilator-free days distribution. So this is what came from the preliminary data in the trial. And again, thinking about this, how we model the data and how we simulate the data really comes from that definition of the outcome. So it has to align with the outcome of interest. So in this case, we're going to simulate the data in each category of this ventilator-free days distribution with a multinomial distribution. So essentially, that means that each patient can only experience one of the categories, and the probability of being in each category is calculated from the data. So there's a much higher probability of being in -1, and then in the larger values for ventilator-free day. And then we evaluate the treatment effects using a proportional odds model. So we have that model, we can set it up, and then to account for heterogeneity across the subgroups, we're going to use dynamic borrowing. So we have a proportional odds treatment effect in both severity states, and we essentially pool or not across those 2 severity states using dynamic borrowing. So that's our model, that's our statistical model for the data and our simulation method for the data. In this case, our trial decision rules, we were interested in superiority and futility. We were only going to evaluate superiority and futility after an initial kind of threshold level of recruitment. So that allows us to make sure that we don't make a kind of erroneous decision really, really early on in the trial, say after 100 patients, because we just had this like people who did really, really well in the intervention arm just in those first few patients. So in this particular case, you can see on the right-hand side that the minimum recruitment level is 750 patients. So that's really kind of our initial recruitment. And then we're going to stop either for superiority, which is the blue box. So if the treatment effect falls with high probability in the blue box, or futility, which is if the treatment effect falls with high probability within the gray box. Again, as I said, the severity states will be evaluated separately from each other, and we're going to have analyses every 3 months after that initial recruitment level. So in terms of the decision rules, we just have kind of the 2, futility and superiority, but we also have this idea that the 2 severity states are going to be separate. And we also need to evaluate what's the appropriate level for the initial recruitment. So we started to think about what our simulation scenarios might be. So we wanted to determine our decision thresholds. What were the best thresholds to make our decisions? And in this particular case, we have three kind of key things that were going to vary in our trial. The probability that we stop for superiority. So essentially what we're doing here is we're calculating the probability of superiority. So the probability that the treatment effect is greater than 1. And then we'll have a threshold that says if that probability is high enough, we'll stop the trial for superiority. So in this case, we compared 4 of these high thresholds, 0.975, 0.99, 9925, and 995. The probability to stop for futility. So again, what we would do is we would calculate the probability of futility. So the probability that you lie within this gray band here, which actually is the treatment effect being less than 1.2. And then if that probability is sufficiently high, we would stop for futility. In this case, we compared 0.85, 0.9, 0.95, and 0.9925. We also had 3 levels for the initial recruitment, 200, 350, 4 levels for the initial recruitment, 200, 350, 500, and 800. And we calculated this with a proportional odds ratio of 1 for the null effect, so when there's no effect of the intervention, and 1.3, which is a positive effect of the intervention. And this actually gives us 128 different scenarios to run, which is where kind of the computational complexity comes in. And then once we decided on some optimal designs, we actually wanted to kind of evaluate this in a lot of different scenarios. So this included a 0.8, which is a scenario where there's harm to the patients in the intervention arm. So really what we wanted to see there was, do we stop the trial almost immediately to avoid harming patients? And then we had a huge range of different proportional odds ratios that represent efficacy of the intervention, just again to see if the effect was smaller than we were anticipating, how likely is it that we would conclude efficacy. And then the other thing that we wanted to compare was settings where the 2 severity states actually differed from each other, and how likely was it that you would conclude superiority for the severity state that was superior when the other one remained neutral. So this was 34 different scenarios. So again, a big simulation burden. So I don't actually have run the simulation in my example, but believe me, we ran the simulation and it took a long time, but we managed to get these results. And so what you can see here, and this is a bit of a messy graph, so I'll just kind of take a bit of time to talk you through it. So this is the power of the trial. So it's the proportion of states where we conclude superiority. And here we have the 2 states separately. And then on the x-axis, we have the expected sample size. So as you'd expect from a kind of standard clinical trial, the higher the sample size, the higher the power. And these scenarios that we have here are only those scenarios where the type 1 error rate was below 5%. So we're not seeing any of the scenarios. So you'll see we did 128 scenarios, but they're not all on this graphic because some of them had unacceptably high type 1 error rates. What you can see in terms of the pattern is that the higher the initial recruitment, so the bigger the symbol, the higher the power, but also the higher the expected sample size, and the superiority probability leads to the higher the superiority probability, the lower the power and the higher the expected sample size. And then similarly for the futility probability, the lower the futility probability, the lower the power, but also with a similar sample size. So really what you're looking for is a kind of trade-off between superiority probability, futility probability, and initial recruitment. And crucially, what you're looking for is you're looking for a design that has low expected sample size and high power. So really we want a design here, the part of the graph that's empty. So we're looking for scenarios that kind of live on this outside edge. And I believe that the scenario, we compared two scenarios. It was this this kind of blue square here, and then this red circle here. So the circle is a slightly lower futility probability, lower superiority probability, and a slightly higher initial recruitment. And then once we'd selected that design, what we actually ended up doing again was comparing it for lots of different odds ratios. So what you actually see is that for the very low odds ratios, so for this harm scenario, for futility, we conclude futility very, very quickly. This is the initial recruitment at 500 patients. And then, so that's great. We know that we can conclude harm quickly. And then for the scenarios with a true odds ratio that's positive, again, for the very high odds ratio, we conclude very quickly superiority, and then that decreases as the odds ratio decreases. So again, we can see from this that if the odds ratio is 1.3, which is what we initially used, there's a high power, but it can take a little bit of time to get to that point. So we need to make sure we have sufficient patients in our trial to be able to conclude. So that is, in a nutshell, some statistical perspectives on designing adaptive platform trials. And really the final piece about how to do those simulations is really, as I say, it's a discussion and a communication between the statistical team and the clinical team to be able to kind of get these plots and decide what's the most appropriate design for your trial and your research question. So I will stop sharing and I will open up for any questions.
All right, if anyone has any questions, please raise your hand or put them in the chat. Richard, I see you have your hand up.
Yeah, thank you so much. Dr. Heath, this was amazing as a relative newcomer to Bayesian statistics, incredibly helpful. So thank you so much for going through all of that. Two questions, both related. I think I have always struggled a bit to understand the concept of dynamic borrowing. And I think my first question is, like, what's actually being borrowed? And if you could maybe talk a little bit about the procedure for that. And then the second question is, maybe I just misunderstood in some like sort of reading and previous sort of review, but I had thought that some of dynamic borrowing meant that you were changing, like upweighting or downweighting the, I guess, weight of the prior probability based on incoming data from sort of the ongoing trial. I just wanted to see if that's sort of not part of the definition or if I just misunderstood that previously.
Okay. Okay, so I think in terms of the first question, this idea of like what's being borrowed is information from patients that are not directly in the group. So whenever we're estimating a treatment effect, essentially what we're doing is we're saying that all these patients are the same, and we're going to estimate the average treatment effect among these patients. That's kind of the goal. And when you do dynamic borrowing, what you're saying is I acknowledge that there's differences between these patients, but that I believe that they're sufficiently similar that some relevant information from those patients can be borrowed to help me estimate the treatment effect for patients A, essentially. So it's information from group A to group B or B to A, based on how closely you believe they are related to each other.
If I might just clarify that, so for a kind of example that we might all be familiar with, in, for example, the DRIVE trial or in the invasive mechanical ventilation domain, are you talking about borrowing information from the low elastance patients to inform the high elastance patients? And would we ever borrow from so that's within a single trial, but we're talking about a platform trial. So would we borrow information from patients who aren't in kind of this 2 comparator arm trial, but are still in the mechanical ventilation domain? And would we even borrow from like the steroid study that's in the practical trial? Like how far do we borrow? Does that make sense?
Yeah, so I think, and actually that kind of feeds really into the other question that you had about, I've seen dynamic borrowing in different contexts. So it can be used in the statistical machinery can be used in lots of different contexts. One context that it can be used in is to say that you have patients who were enrolled in a specific time point, and I have data from patients who were enrolled before that time point, and I'm going to borrow from those patients from before into now. And it's from like a statistical perspective, that's the same, exactly the same machinery, but conceptually, like how you might clinically, how you might argue that that's relevant would have to be from a slightly different perspective. So maybe you'd say that like we don't think there's a significant time trend in this particular intervention because the standard of care is very protocolized. Then you could say, okay, we can borrow from prior to into the trial. But if you had big changes in standard of care, you might say, oh, well, they're not relevant. Same. So for elastance, you're essentially making an argument that you think that there are some similarities between the treatment effect in these two patient populations. So that might be because you're saying that the mechanism of action is the same. So if the mechanism of action works, we expect it to work in both. But because they have different baseline risks, maybe we're not expecting the treatment effect to be exactly the same. And that's why you allow yourself those differences. So how far you can take it really, to my mind, is like a clinical call. It's like, how do you think these patients are similar enough that they will provide information about each other? And then on the other end of the spectrum, do you think that these patients are so similar that there's no point in accounting for the differences? And so it's really when you land in between those two goalposts of like, these patients are completely separate and these patients are exactly the same, that's where you really would start to think about borrowing. And actually, like, it's not necessarily, I don't think there's great, I mean, like a plug for some research that we're doing right now is I don't think we have a good idea of how to turn that clinical understanding of how similar these patients are into something that we can use in the analysis models. And that's something that we're hoping to work on over the next year. I've got a brilliant PhD student who's hoping to do it, so that we can kind of actually use that knowledge and really, I think, bridge between this like borrowing thing that we talk about and actually how we can use your clinical expertise and experience to help do that.
Great. Thanks so much for the explanation. That was perfect. Thank you.
Thanks, Richard. Any other questions? And I had a question while other people are mulling over how to frame their own questions. With this sequential design adaptive approach, one thing we hear in frequentist design trials is that we're spending our power every time we take an interim look. So can you explain like why that isn't an issue with this approach?
Yes. Can I explain it clearly and concisely? I don't know. So it's actually a philosophical difference, and I think that's really where I guess the challenge is that like statistical philosophy might not be like the most exciting thing to discuss, but essentially the philosophy that underpins the Bayesian methodology means that when you, sorry, let me go the other way. When you analyze when you analyze data in a frequentist context, the way that you interpret it is basically by saying, if I was to run this trial again and again and again and again, what is the chance that this outcome occurred? And so because that's how they define probability, the way that they run the trial matters, essentially. Like the interpretation of probability relies on you understanding the conduct of the trial. So when you're thinking about an adaptive trial and you say, okay, well, if I run this adaptive trial with this design over and over again, then essentially that's where you're spending the type 1 error because you're kind of doing multiple analyses. A Bayesian interpretation of probability doesn't rely on understanding how the trial would have been done if you had run the trial to completion or something like that. It just relies on the data itself. So what data you have collected. And so that's why we don't have to worry so much about the spending or that kind of thing, because our interpretation of probability doesn't require that we understand how the trial works. There's a really great paper about which says that basically like the way that we get away with not being able to kind of ignore the way that the data was collected is we have a prior distribution. And so either you have to like conceptualize probability as understanding how the trial works and doing an infinity of trials, or you accept that you need a prior distribution. And then by having that, we allow ourselves to like get away from all that complexity. So I think that's the philosophical answer. In practice, we actually do calculate the frequentist operating characteristics of our trials. And so that we do kind of have that same. Sometimes, if we were to say, let's do an analysis every patient, that actually would make it kind of harder for us to design our trial with respect to those frequentist characteristics. But a Bayesian, like a pure Bayesian who doesn't care about frequentist operating characteristics like power would be like, it doesn't matter, I don't care.
Kathy?
Yeah, thanks, Anna, for this excellent talk. I have a question for you. It's curious to me to understand, like with the integration of AI, do you think that that will help ease like the planning of statistical analysis for adaptive platform trial, or would it be more challenging? What are your thoughts on that?
I think so. I guess there's two answers. So some of the work that myself and my colleagues have been doing is trying to reduce the burden of these simulations using like machine learning models and things like that to kind of understand. So in that sense, there's sort of methodology in the pipeline to make it easier to do these designs using those tools. But in terms of maybe more the like generative AI, like, large language models, my gut is that you at the moment, I think you still have to run the simulations. Certainly they can help with like coding up the simulations. We've used it in our team to kind of find ways to do things more computationally efficiently, or like write our code in a different language that's easier to run and stuff like that. So that's really helped kind of overcome some of those challenges. For the moment, I still think that there needs to be a person doing it, but I don't know if we know what's coming down the pipeline to take our jobs away. So I don't know. I hope that there's still scope for us to do this stuff, but it may also be a really helpful tool for communication. Like I think there's one of the challenges for statisticians, I think, is that we're not often trained very well to communicate. And so I think having tools that help you communicate with your collaborators will only help us kind of translate between that mathematical language that we're maybe more comfortable speaking and a kind of more lay language that you need to be able to actually do this work in practice.
Thank you.
Great.
Thank you.
Thanks, Anna. I have a million more questions to ask you, I'm sure, but it is 5:01, so we will go ahead and end the webinar. And I just want to thank you and Kathy and everyone for joining us today. So thank you so much.
Thank you.